So as we've been learning about DNA, there are only 4 bases that you can possibly use. You only have 4 bases, and those 4 bases specify only 20 amino acids. So if you think about that, or if you think about the fact that there are only 4, you can't just have one base specify an amino acid, right? It doesn't make any sense. You only have 4 bases, but you have 20 amino acids that you need to talk about. And then you can't just use 2 DNA bases because there's too many amino acids. If you only use 2, you would only be able to specify 16 combinations. So the reality of what happens, as implied by this person, Sidney Brenner, is that there is a triplet code. Okay, 3 DNA base pairs are used to specify 1 amino acid. 3 DNA base pairs are used to specify 1 amino acid, which gives you 64 possible combinations. Okay? You have 4 letters possible, you have 3 positions, you have 64 different combinations to specify the 20 different amino acids. And so, like I was saying, you have 3 bases here. So here's your DNA, here's your RNA transcript, and this CGU specifies arginine, and UCA specifies serine, and UGG specifies tryptophan, and ACU specifies threonine. So you have 3 letters of DNA specifying each one amino acid. So which 3 bases specify which amino acid, and why is it that there's extra information that you're just leaving here? You can specify 64 different amino acids. Why are we using 3 base pairs for this? And the reason for this is because the code is redundant. There are different 3 base pair codes that can specify the same exact amino acid. And you need spots for starting and stopping as well. Okay, you need to be able to tell your ribosome, okay, start building this protein here and stop building this protein here. Interestingly, the genetic code is universal, so it's the same for bacteria as it is for humans. And that's pretty great evidence, not that we needed any more evidence, but it's pretty great evidence for evolution. It's pretty great evidence to say that we came from the same place. Now, this isn't to say that bacteria and humans always use the same exact letters. Different species have different 3-letter codes that they prefer to use, but you can make this work in any living organism on Earth. The 3-letter code works for any living organism on Earth to specify amino acids. So this is a chart that shows you how to read which set of base pairs is going to turn into which amino acid, and this is written in RNA. You can tell it's written in RNA because there are U's instead of T's. But let's say we wanted to see what, for instance, ACG looked like. We want to know what ACG, we want to know which amino acid is specified by ACG. So we go over here on the left, and we find our first letter, A. The next thing you want to do after you found your first letter is go to your second letter, which is C. You see C up here. So we're right here, AC, and then our final letter is G, which is threonine. ACG specifies threonine, THR. And so the reason that it's important for this genetic code to be redundant is because if you have a mutation, let's say our G accidentally gets turned into an A, you still have the same amino acid. So even though you had what seems like could have been a disastrous mutation here, you're still specifying the exact same amino acid, and actually nothing changed. These are those silent mutations I was talking about the other day in one of the previous lectures. These are silent mutations. They didn't change anything at all. So even though ACG and ACC are different base pairs, they're different DNA codes, they're still specifying the exact same amino acid, which is a big deal That is the reason why you have 64 possible combinations of those 4 letters, and you only have 20 amino acids that you need to specify. It's a protective measure so that things don't get messed up. So the next video, we're going to start talking about translation, the final step in building a protein.